Biotechnology is genetically changing microorganisms to produce drugs, vaccines, and other cures. It also includes changing biological processes to achieve a certain result. One large category of biotechnology is GMOs. These are genetically modified organisms, and they can be made by taking the desired gene from one organism and putting it in another organism. This is an example of genetic engineering. Genetic engineering is modifying the characteristics of an organism by manipulating its genes. Scientists can also use gene manipulation to create pharmaceutical products such as insulin. The process starts with a specific strand of DNA from an organism. Restriction enzymes then come and remove a gene. This gene is replaced by another gene with a desired trait. The new gene is attached to the DNA strand by DNA ligase. This is how recombinant DNA is formed. A lot of DNA is required for genetic engineering, so scientists use DNA cloning, aka gene cloning. This replicates pieces of DNA. During gene cloning, a plasmid, which is a circular DNA strand, is removed from a bacteria. The restriction enzymes cut out a gene, and a gene from another organism replaces it. This is a recombinant plasmid. The recombinant plasmid is placed inside bacteria to create a recombinant bacteria. The recombinant bacteria then multiplies to create more of the desired DNA. An alternative to DNA cloning is a polymerase chain reaction. This process uses primers, which are short single-strand DNA fragments, and it also uses a DNA polymerase. The first step is denaturation, in which the complementary strands of DNA are broken. The next step is annealing, in which the primers attach to the separated DNA strands. The final step is extension, in which the temperature is raised and the new DNA strands are made. This process then repeats. The results of PCR are visualized through gel electrophoresis. In this process, fragments of DNA are pulled through an electric current, which separates them according to size. Another part of biotechnology is cloning. There are two main types of cloning: organismal cloning and therapeutic cloning. We are focusing on organismal cloning. This process uses adult stem cells, which are undifferentiated cells in the body, and also uses embryonic stem cells. A specific example of organismal cloning is somatic cell nuclear transfer. In this process, the DNA is removed from a donated egg. An adult cell is taken from the organism being cloned. DNA is taken from the cell's nucleus and put into the donated egg's nucleus. The newly modified egg is then artificially induced to create an embryo and is placed in the uterine environment. The normal development then occurs and a clone is born. One last part of biotechnology is the synthesis of small molecules to use for drugs. This includes all processes that involve chemical reactions that create helpful drugs. Biotechnology is important to increase lifespan and to improve the quality of our lives. Biotechnology improves foods, cures diseases, helps make new organs, and saves lives.